Dr. Tony Moody. He is at the Duke University, and he's on the faculty of the Duke Human Vaccine Institute. He's also one of the civic vaccine centers he's one of my counterparts at Duke, running the vaccine preclinical studies there. And so I will turn it over to you, Tony, and let you introduce the next.
Thank you, Ted. Thanks, Ted. Much appreciated, and I'm glad to be a part of this program today. So it's my honor to introduce Dr. Andreas Handel, who is at the University of Georgia in the College of Public Health, and also the Associate Department Head and Graduate Coordinator of the Health Systems Modeling Core at UGA. So he's going to talk to us about the role of influenza vaccine dose toward homologous and heterologous protection.
Thanks for the introduction. Next slide, please. Okay, so I want to discuss a bit the role of dose, and I think it's generally appreciated that dose, either in a natural infection or in vaccines, often plays an important role. One criteria, or one important one, is getting exposed to a certain dose, the probability of getting infected. So, we quantify that by the ID50. Then given infection, dose can still often play an important role. So higher dose can lead to more severe outcomes if it's a natural infection with influenza or other pathogens. In the lab, we often quantify that by the LD50 if it's a very severe pathogen. And then for vaccines, which I want to focus here, it's generally thought that an increase in dose might lead to better immune response and better protection, but also probably will increase side effects. So in the figure that I drew here, that schematic, often we think of it maybe in something is either saturating, so at lower levels not much, and then it ramps up, and then the dose gets really high, it saturates again, or it could be non-saturating like that straight line I drew, or even accelerating. Next slide, please. And so the current approach to picking a dose for any kind of vaccine happens usually during phase 1 and phase 2 trials. A few doses, usually around 3, 2 to 4 or so, are being tried based on thoughts or data that came out of animal studies. And then based on those, a dose is chosen in what I consider a not too rigorous manner. I mean, the data is looked at and then looked at the immunogenicity and potential side effects, and then a dose that looks sort of overall the best in the assessment of the investigators is chosen. So I just put here a slide from the Pfizer-BioNTech COVID vaccine. You saw here they had 5 different doses, though one dose was the highest dose was in a non-boost and the other one were boost. And so, basically based on this data and also the side effects, they then decide, okay, let's go ahead with the 30 microgram, which is then the one that was moved into phase 3 trials. Next slide, please. That usually works okay, and we have good vaccines for a lot of diseases, but I think it's not quite optimal. So there's several issues that can be important. So one is that the immunogenicity might actually not be monotone. And an interesting recent example might be the AstraZeneca COVID vaccine. I'm sure all of you heard in the news that they accidentally gave a group half a dose in the first shot, and that seemed to have a higher immunogenicity. So I know what it all means is unclear, because it seems like that subgroup was different than the others. So it's not quite clear, but there seems to be indications, or at least possibilities, that as you increase the dose, it might at some point actually the protection gets worse. And another important consideration, especially for flu, and I'll discuss that a little bit here, is we also want to not just introduce a strong response against the vaccine strains, but we want to have broad protection against all kinds of strains. And again, this could be a monotone increasing, or it could be a peaked response where the optimum is actually somewhere in the middle. Next slide, please. So the overall sort of goal of what I've been thinking about and working on for a few years is see if we can come up with fairly detailed understanding and models that would allow us to use the few doses that are being used in phase 1 and 2 trials, and we can't do more because every dose is more expensive, so there's a limit to that, but use that somewhat sparse data and maybe combine it with some analysis and modeling that could further help us tweak the dose and optimize it. I put a few references up there, and if you choose to get the slides from my web page, you can just click on that and get to the actual papers. The first is work that we've done in our group, an early exploration, just sort of a conceptual, and then a more recent one where we looked at some infection studies. These were animal data, and calibrated a model to it and then ran that to try to predict what would happen for all kinds of doses. And the figure you see at the bottom is actually a prediction from that model. So in that specific instance, you see that actually the optimum is somewhere in the middle. And once you exceed a certain dose, it actually gets worse in terms of protection. Interestingly, morbidity goes down there in this model as well. Now, this is very conceptual, so I wouldn't take that too serious at this stage. I put links to 2 other references as well. Sophie Rhodes in the UK with Richard White and others have also done really nice work. The first paper is one where they looked at a TB vaccine candidate and data from animal studies there, and they also saw an indication that maybe there's a peak response, and as you go higher, the protection goes down. And the second paper is a nice recent sort of commentary opinion piece that they wrote. Next slide, please. So after that sort of introduction, I want to briefly discuss a recent project we are close to wrapping up. And this uses Ted's flu cohort data that was already mentioned. And specifically, we're looking at antibody responses in individuals that got the seasonal flu vaccine. So this is not a clinical trial. This is just individuals that choose to come and get the flu vaccine. This was from 2014-15 season to the 18-19 season. The study is ongoing, but the data we analyzed here ran up to the 18-19 season. And so the default vaccine was the regular standard dose Fluzone vaccine, but then individuals that were older than 65 were offered to receive the high dose vaccine. So unfortunately, we have only 2 doses, but it's better than nothing. So we were interested in seeing if we can find differences there. Next slide, please. So the overall question is, is there a difference between standard dose and high dose vaccine on immune outcomes? And more specifically, we looked at antibody titers measured by HAI. We looked at 3 different outcomes. They're all sort of measuring the same thing, but are slightly different. One is the fold change in titer before and after vaccination. Then is seroconversion of individuals and seroprotection. And since only the older individuals got the high dose vaccine, we decided to also limit us only to those individuals in the standard dose. So the ages were somewhat comparable because we already know that age is an important factor and could influence results. Next slide, please. So here's an example of the kind of analysis we did. So during those years, there were 2 different H1N1 strains in the vaccine, the California 09 and the Michigan 15. And so what you see here is the fold change for those that got the high dose and those that got the standard dose, and then the ratio of the 2. So basically, if the high dose vaccine did not have any induced immune response, not any better than the standard dose, then the ratio would be at 1. Here you see it's away from 1, so it seems to indicate that the high dose vaccine actually induced higher levels of antibodies, both for the California and Michigan. And all the analysis I'll show you is look the same. It's basically the median and 95% credible intervals here. You see it's a pretty widespread, even though sample sizes are decent. The little number you see, 297 and 57 is the sample size, but there's just a lot of heterogeneity because it's not a controlled trial. It's natural people that chose to participate in this observational study. Next slide, please. Here's another outcome, the seroconversion. So again, we looked at seroconversion in those with the standard dose and the high dose, and then looked at the difference. So if the high dose vaccine were not any better, we would sit at the 0 mark. But here there's more seroconversion in those that received the high dose. Next slide, please. And similar for seroprotection. In this case, you see there seems to be an increased protection for those receiving the high-dose vaccine. Next slide, please. Then we looked at the H3N2 component, and there it's not as clear. So if you look at the first two, the Texas and Switzerland vaccine strains, you see they're very close to zero, meaning that the high-dose vaccine actually didn't induce more or a higher HAI titer. And in fact, the median for the Texas is somewhat below 1, so the median actually led to a decrease in the high dose, though it not very much overlaps 1, so I would interpret that there's really no difference between the high and low dose. Well, for the Hong Kong 2014, again, there seems to be a benefit to the high dose vaccine. Next slide, please. This shows the same, just again, seroconversion, similar patterns seen here. Next slide, please. And seroprotection seems slightly different. There is not much of a difference between those 3 strains. Next slide, please. So then one more on the B component. The high dose only has 1 component, so we only looked at that one that was actually in the high dose vaccine and compared that to the standard dose. And so there were 3 B strains during those years where the data was collected. And you see overall, it seems to suggest that at least for the HI ratios and the seroconversion, the high dose seems to help. Seroprotection, the Brisbane is actually on the negative side. But again, it's pretty far overlapping. Next slide. So what I've showed you so far was a univariate analysis where we just looked at the dose and the outcome. We had some additional information on those individuals. We had, of course, their pre-existing titer. Now, if we do fold, it's already kind of factored in, but it could still play a role. So we decided to do a multivariate model where we included that, also gender, age, and race. That was kind of the maximum data we had available that we could include. What you'll see, I'm just showing one example, but overall you see very similar results. On the top is the univariate, and then the bottom, just for comparison, the multivariate. So things shift around a little bit, but the overall patterns look the same. And we also did a different approach called a propensity score matching, where you try to pick individuals in the standard dose that are very similar to the high dose group. And again, the overall patterns we see are very similar. Next slide, please. Now, the nice thing in this data set is that we not only have HII to the vaccine strain, but there's a whole panel of historic strain that Ted and his group ran. So we could look at the difference between high dose and low dose, not only against the actual vaccine strain, but sort of look an indication of breadth of protection. So here's an example for the H1N1 components. And so each column is the vaccine strain. So the first 2, basically the diagonal would be what I showed you earlier, the strain with itself, the vaccine strain, and all the other ones are then basically looking at cross protection. And you see overall here, it seems to indicate that high dose is beneficial, though it's not that strong in effect. And sometimes you see it even on the other side, meaning that the median is negative. Next slide, please. Similar for the H3N2. Again, overall, there's sort of more green than red. So overall, it seems to indicate that it's positive, but it doesn't seem much of an effect, apart from the occasional strain where it's pretty far away from sort of the null mark, the 1. I'm just showing here the ratio, but the seroconversion and seroprotection look overall fairly similar. Next slide, please. And then we decided also to look at stratified by gender, because it's known that gender could have an impact, and we thought maybe there's actually something to see if we look at gender and dose. And there's some interesting patterns. If you look, for instance, for the Switzerland, the middle column, there seems to be fairly consistently that the high dose is beneficial for females, but not for males. So the circles tend to be shifted towards the left, and the median seems to be actually below. So it means it's not helpful, while the squares are on the other side. If you look at the other 2 vaccine strains, that pattern does not seem to be there. So it's an interesting finding. It's worth following up further that there might occasionally be interactions between gender and strain, but it's something we definitely need to look in more detail, and it doesn't seem to be consistent. I'm just showing H3N2 here. In the H1N1, we didn't see a striking one for the 2 vaccine strains there. Next slide. Okay, and that wraps it up. So on this analysis, I think overall one can conclude that the higher dose does seem to induce antibodies more definitely against the vaccine strain, though it can depend, and for some years it doesn't seem to actually do much. It seems it's very limited. We only have 2 H1 vaccine strains and 3 H3, so it's somewhat limited, but it seems to be based on that limited data benefit H3N2 less than H1 and B. Similar for cross protection, it seems to be that the high dose is overall better, but the effect doesn't seem to be very strong and somewhat inconsistent. And then, as I mentioned, this may be interesting interaction between dose and gender. I want to reiterate that this is an exploratory analysis, so we basically went in and just sliced and diced the data. So this definitely needs further investigation on confirmation. But I think it's worth looking in more and trying to further understand what is the role of dose and how do we optimize future universal flu vaccines with regard to dose so they're as protective as possible, both against the vaccine, but then of course we want broad protection from a universal vaccine. Next slide, please. And that's my wrap-up slide. I want to thank the collaborators on this. The group members Yang Yi and Brian McKay have worked on this, and then a lot of the civic members, especially Yishan and Ted Ross. And again, if you want the slides, you can get them from that webpage. And if there's time for questions, I'd be happy to take any.
Yes, there is time for questions. Sorry about the next here. Let's see. So first question from Gerald Reicher, which is sort of an odd question. Can the nano-flu vaccine and COVID vaccine be successfully combined into one vaccine? Not sure it's directly relevant to your talk, but it'd be interesting to get your opinion on that.
No, I don't know. I hope so. I think it might be. I don't see why not, but I haven't seen any data one way or another, so I feel like it's quite speculative at this point.
Yeah, that's sort of my take on it as well, because the challenge is that you have to do all of the various combination studies and trials, and so it will be complicated. Elena Vasilyeva, sorry if I mispronounced the name. Do you have an opinion about the best dose correlation of human dose for mouse experiments for flu vaccines, single vaccination?
If I understand it right, the question is if you have animal data, can you move to human data? That's interesting. I've been thinking about that. I don't have any clear answer yet, but I know that the colleagues Sophie Rhodes that I mentioned earlier, they have been working and thinking about that a little bit too. And there are, I think I've seen at least in one of their studies, I don't know if it's published yet, where they tried some kind of allometric scaling, basically saying, okay, you know, if a mouse is that big and a human is that big, and if the peak is after 2 days in a mouse and after 5 days in a human, how can we go there? I think we don't have a rigorous way of going from one to the other yet. However, I think it could be quite interesting. I don't know if mouse to human might be a big jump, but maybe ferret to human might be a much more palatable jump that I think would be worth exploring.
Fantastic. And I'll take chair's prerogative. I have a question for you. So I'm a pediatrician, and I'm very comfortable with the idea of weight-based dosing and thinking about age, taking age into account, you know, with different age ranges, having different dosing guidelines. And then, of course, there are plenty of data in adults and children as well, showing that malnutrition, both, you know, either undernutrition or overnutrition and obesity, can reduce vaccine response. Do you anticipate getting to a point where we'll need to think about vaccine dosing in a more complicated way than we're doing currently?
I think it is worth doing. It seems like one step at a time. Right now, we don't even have sort of a rigorous way of picking a dose for the average human, but I think as we go forward, that's definitely worth thinking about. And the nice thing is that In recent years, the data that Ted has been collecting actually does have BMI in it, so that's one thing we definitely would like to look at. In this study, we combined some older data where we didn't have that information, so we couldn't look for that, but it's definitely on the list to look at in future studies. And also, other factors that could be looked at. At that point, it might actually be also useful to try to use these challenge study data or similar ones, because they are somewhat more controlled and sometimes doses are higher. So that could get us a bit further as well.
That makes sense. Fantastic. I don't see any more questions, so I think we'll go ahead and move on to the next presentation. I have the honor of introducing Nick Heaton, who is also here with me at Duke University and a member of our Duke Civic Vaccine Center. And he is going to speak to us about viral genetic approaches for developing improved influenza virus vaccines. So take it away, Nick.
Thank you, Tony. Thanks for the introduction. And it's my pleasure to be here with you all today, if only virtually. As Tony mentioned, I'm going to give at a reasonably high level an overview of some of the concepts and some of the approaches that our group is taking, trying to really build influenza viruses that elicit different and better immune responses after vaccination. Next slide. So if there's an overall kind of thesis underlying the approach that we've taken, it's that while there's certainly limitations to current influenza vaccines, there's also some good characteristics about them. Namely, they're immunogenic and safe, and actually reasonably protective against infection with matched strains. Advance, please. And the goal of what I'm going to talk about today is essentially how do we save what's good about vaccines with respect to better vaccines and then make them better. So can we take what we already like and improve upon it, as opposed to start from scratch with a completely new approach to generate kind of next generation vaccines. Advance. So this is the influenza virus particle, and again, the approaches that I'm going to talk about today are all with respect to altering this kind of authentic virus particle to fundamentally elicit different immune responses. And so when we think about wanting to do that, the first thing we need to understand is what we want to do. How do we want to change this thing, and what's the basis for making those changes? Or another way to say that is really we need to understand what the relative contributions of these different virion components are. And what I'm going to talk about today is with respect to humoral protection of the eliciting of antibodies. Advance, please. So as you've heard and as you all know, the hemagglutinin, which is one of the surface glycoproteins, is the antigen and the primary target, certainly for eliciting neutralizing immunity. Antibody responses against the neuraminidase can also be protective, but the way that we test kind of the contributions of these viral proteins, or at least predominantly have in the past, is in isolation. So below the virion, I'm just showing you a hemagglutinin and neuraminidase, and typically how we understand if viral proteins or components of the virion contribute to protection is to take one out and vaccinate just with that and understand the type of immune responses we get. But again, if our goal is to vaccinate with this whole virus particle, what we really want to know are the aggregate contributions together of a subset of viral proteins. And so the idea that we had in some data that I'm going to show you now is really based on the idea of a subtractive analysis. Instead of taking one virus protein out and asking what it can do with respect to eliciting immune responses, what if we take a whole virus particle and start deleting proteins, again, while maintaining the structure, and the genomic content and all these things of the actual virus particle. And the data I'm going to show you today is doing that while by deleting the hemagglutinin. So if we get rid of that major antigen, what do all the other proteins do? Advance the slide, please. So on the left, first I'm showing you, a scheme of how a virus is normally amplified. So if you have a wild type virus, it encodes all the information it needs to replicate itself. It can infect cells like MDCK cells, which are highly permissive to flu. It can infect cells like T cells, which are highly permissive to viruses. And then it and you get copies of that virus out, which you can then collect and use for vaccination. Advance, please. The first step to us kind of generating our -1 protein virus particles is to genetically delete the hemagglutinin. So the hemagglutinin is encoded in segment 4 in the genome. And what we've done in this case is delete the open reading frame for the hemagglutinin and replace it with M-cherry as just a fluorescent protein for ease of detection. But we've kept the RNA motifs that allow the virus to see that segment. So the virus still thinks it has hemagglutinin. And this virus wouldn't be viable. It doesn't encode the receptor that it needs. But if you propagate this virus on cells which already have the hemagglutinin, which I'm diagramming in the middle here, this virus replicates just fine. Instead of the virus encoding its own protein, just gets it from the cells, and you can amplify this virus and grow it to high titers. But again, the virus particle that comes out of that still has the hemagglutinin. Vaccinate with that and ask our question. But we can take this deletion virus and then infect a new cell line, a cell line that doesn't have the hemagglutinin. It turns out, and there's a number of papers that show this, that the hemagglutinin itself is important for viral assembly and budding. So what we did was take the transmembrane domain and cytoplasmic tail, which are just a few amino acids, and make a cell line that harbors that, but all of the ectodomain, everything that's relevant for the hemagglutinin really in terms of eliciting immune responses is gone. It's been replaced with GFP again, just so that we can see it. So now the virus will be able to affect these cells. So we take the hemagglutinin containing virus particle that's deleted for the ORF. We infect these cells, and what we get out is really an authentic virus. It has all of the viral proteins that it would normally have. It has all the RNA and ribonucleic complexes. And the hemagglutinins been replaced by GFP. So we can vaccinate with this virus and the control virus and understand kind of what the contributions are from the hemagglutinin plus everything else and just the everything else itself. Next slide, please. So on the left again is the scheme that I'm showing you that I just showed you, and on the top is a picture of it working. So the top right, the GFP, the cell line has GFP, right? It's fused to this transmembrane domain of the hemagglutinin, so it's at the plasma membrane. If we infect with this virus, it has M-cherry instead of the HA ORF, the infected cells are red. And then what we can do is collect those virions and understand what their composition is, and I'm showing you that on the bottom right. So on the left of the bottom right, you can see if we blot for the hemagglutinin, the wild type controls have a lot of hemagglutinin. There's no detectable HA in our deletion virus, which is good. This is normalized to the matrix protein, M1, which is kind of the structural component of the virus, which is inside the viral envelope. GFP is only in the HA negative, which is good. And if you look at the other two kind of surface exposed proteins in the virus particle, M2 and neuraminidase, they're reasonably similar, if anything, a little higher in the HA deletion virus. So it seems like we have the proteins there that we want or don't want. So what happens if we vaccinate? Next slide, please. So this is the data I'm going to show you is from this scheme. It's diagrammed on the top. It's a single prime boost with no adjuvant. And then we can do challenge and various other analyses, which I'll show you over the subsequent slides. If you look below that, what I'm showing you is ELISA reactivity against different kind of key viral proteins. So on the left is the hemagglutinin, and I should have said this virus strain was generated in a laboratory adapted H1N1 background, which is good for mouse challenge studies. And so we vaccinate mice with that strain, and then we take out the serum. We're looking for reactivity against the matched hemagglutinin. And you can see that the wild type virus is in purple. We get strong HA responses, but in blue is our HA negative response, and we get no appreciable response, very similar to our controls. But in the middle is the response against the neuraminidase, which again, we had approximately equal level of neuraminidase between our wild type and our HA negative virus. You can see the results. In the middle, we get good reactivity, neuraminidase there. And if you do an ELISA against a whole virus, you can see it's basically the average of those two graphs on the left. you get more reactivity against the wild type because you have the contribution of the hemagglutinin. So that's kind of what the CRTG binding profile looks like, but are these things functional? Does it protect from challenge? Next slide, please. So if you look at the challenge data that is shown on the top, and I'll point out that at this dose, all of the control animals, the BSA kind of irrelevant control vaccine animals succumbed to infection, but all of the vaccinated animals survived. You can see there's a body weight dip on the top left with the HA negative vaccine, right? They weren't protected as well. Animals weren't protected as well if they got a vaccine with no hemagglutinin, but ultimately all recovered and survived. You can see the lung histology on the bottom left. Again, uninfected is what the lungs should look like. BSA vaccine, these lungs are in rough shape. There's a ton of immune infiltration. The vaccine groups all look reasonably similar to the uninfected. But on the bottom right is what I really want to point out here, is that this kind of similar level of protection with respect to the vaccine did not give a similar level of reduction in viral titer. So again, in the control, the virus replicates to incredibly high levels, more than 10 to the 7 PFU per mil of lung homogenate. The wild type vaccine, which had hemagglutinin, there's no detectable virus in those lungs at all. For the HA negative vaccine, which again is on the right, significantly reduced virus titer by a couple orders of magnitude, but still dramatically more virus than the vaccine that has the hemagglutinin. Next slide. But again, all of that was with a homologous challenge, right? So the exact same challenge strain as what was vaccinated with. What about a heterologous challenge? And I'm going to show you data for a homosubtypic but heterologous challenge. A strain from 2009, H1N1 strain from 2009. PR8 is from 1934. Next slide, please. So again, the same vaccination scheme. They're vaccinated with this PR8 derived vaccine, which either has hemagglutinin or doesn't. Virus particles, which either have a hemagglutinin or doesn't. What you can see now again is all the control animals succumb to the challenge. All of the vaccinated groups survive, and now there's no difference in morbidity or mortality. On the bottom left, you can see the lungs. Again, there's no difference. The control or the vaccinated animals are protected. And then if you look in the viral titer, which is on the bottom right, now we've lost kind of this effect in differential suppression of virus. And this makes sense because the hemagglutinin is highly divergent between PR8 and CAL09. So it makes sense that the antibody responses to the hemagglutinin are going to play less of an important role here. But I think the important takeaway from this is that the other components of the virion itself, which are more conserved than the surface glycoproteins, are sufficient to protect from disease, even with this, highly challenge from this highly divergent strain. Next slide, please. So at least from the data that I've shown you, we can separate kind of total virion-directed immunity versus everything that's not HA-directed immunity. Right, and the top point is that HA responses are clearly driving control of infectious virus burden, but the immunity to these non-HA proteins are protecting from disease even when viral titer is less well controlled. At least 3 logs, kind of higher viral titer, are observed, but the same, at the end of the day, basic protection from the disease. So, this data is leading us to think about potentially different goals for vaccines. And again, back to this idea of building viruses. Maybe if we build viruses that are designed to relatively increase non-HA immunity, right? De-emphasize the HA immune responses, would this increase the breadth of protection from viral disease? Again, we would expect such vaccines may not have strong neutralizing ability, but this increased breadth may allow kind of a more universal protection. You can also envision a scenario, which I'm talking about at the point below that, where you want to go the opposite way. Maybe we really want to double down on this HA-based immunity to form a more sterilizing vaccine. This would be in a case where we know the exact virus that we want to vaccinate against, like an event of a pandemic outbreak, for example, where reducing virus titers will be particularly important potentially for reducing transmission, right? So anyway, these are just two examples, but the idea is, you know, we may want to build viruses that go different ways in terms of kind of tailoring the immune response. But how do we go about doing that? The next slide. And again, our approach is trying to change these virus particles to alter immune responses across these virus proteins, and we take a genetic approach. And I'm going to tell you about two approaches in the remainder of the talk. The one on the left is a genetic approach to alter HA expression. So we end up reducing HA content in the virion to increase responses against the other virus proteins. And on the right is rearranging the viral genome to make more hemagglutinin and pack more hemagglutinin onto that virus particle to increase relative HA immunity. Next slide. Okay, so again, the first approach, this is the idea of increasing kind of a virus that generates responses that are going to be more broadly protective, right? We're going to de-emphasize the hemagglutinin here to try and get more responses against the other components of the virion, which we think will be more protective against heterologous virus challenge. So the virus is diagrammed on the left. All the viral proteins are encoded for in the genome here, and I mentioned this before, the hemagglutinin is encoded in segment 4. There's these regions on the 5' and 3' end of that genomic segment that are responsible for how efficiently that segment is translated. The RNAs derived from that segment or transcribed from that segment are translated. And our lab has mapped these regions, and we can put mutations in them, mutations that don't change the sequence of the HA that's subsequently made, but changes how much of it is made. So if we generate these viruses and use it to infect cells and collect those virus particles, that's what I'm showing you on the right, is a Western blot from concentrated virus particles. Again, M1 is the matrix protein, which is kind of the loading control. You can see we can get dramatically less hemagglutinin, kind of pound for pound in a virion prep. But now what does this mean if we vaccinate, right? Or can we actually accomplish our goal of changing relative immune responses? And to convince you that we're not the only ones interested in such questions, next slide. I'm going to show you some data from a different group. This is from Peter Palesi's lab, which took a different approach to change the relative expression of the virus glycoproteins. But the data are similar to ours. I'm going to show you theirs now. So on the bottom left is the seroreactivity after vaccination. So again, they made a virus that had less hemagglutinin, more neuraminidase. And when you vaccinate, you can see higher neuraminidase responses, lower hemagglutinin responses, as you would hope for. And on the right is actually a representative graph of a challenge with a heterologous strain. So you actually get more protection when you do a heterologous challenge if you're able to kind of de-emphasize, make a virus genetically that elicits a response, which de-emphasizes the hemagglutinin response and then helps kind of redistribute the immune response to non-HA antigens. Again, this data is similar to what we've seen. So it's a good kind of proof of concept that we can do this. We can build, again, kind of authentic virus particles, which are present in these antigens in the context that they would be, kind of naturally encountered, and fundamentally change the immune response that's subsequently elicited. Next slide. Right, but that was only half of the equation, right? The approach that I just talked about may be useful for increasing the breadth of protection, but what about doubling down on HA and increasing HA responses to really try and make a specific and strong response to a particular hemagglutinin? The approach that I'm showing you on the left is a genomic rearrangement approach that our lab developed, and I don't have time to get into the details. But suffice to say, what we did was move the neuraminidase. So the neuraminidase is normally encoded in segment 6, and we moved it into segment 4 with the hemagglutinin that's normally encoded. And again, the details aren't particularly important for the talk, but at the end of the day, when the proteins are expressed from this segment, they're processed and trafficked appropriately. They're not fusion proteins, they're 2 separate proteins. By the time they get to the plasma membrane, which is where they're going, they're completely separate and wild type functional proteins. And what that allows us to do is then where the neuraminidase used to be, which is kind of this green segment in the virus, we can put another hemagglutinin. Would probably put a second copy of the same hemagglutinin we were interested in, but in the data I'll show you, we put a different hemagglutinin, which facilitates our ability to measure the responses to that hemagglutinin and understand the efficacy of antibodies elicited against it, which is just the point of doing that. So if you generate this virus, what do you get? And the answer is that you get really a 2x hemagglutinin containing virus particle. So on the top right, I'm showing you a Western blot where I'm comparing the dual HA virus or the bivalent virus to a parental H1N1 virus. And you can see that there's approximately the same amount of hemagglutinin as the parent, which only has 1 hemagglutinin. And on the bottom is the H3 parent. And again, we're getting as much hemagglutinin on the dual HA virus particle normalized to the structural protein M1. So this is a virus particle which now just has twice as much hemagglutinin. Next slide. If we vaccinate with this, we get twice as much HA response. So on the left, I'm showing you an ELISA against an H1N1 hemagglutinin. Again, if we vaccinate with the parental H1N1 in black, we get strong responses. If we vaccinate with the virus that has dual HA in green, we get as good or better of a response. The H3 virus in red and the mock vaccinated in blue, there's no response. But we get kind of the reciprocal response on the right if we look for reactivity against the H3. Again, our dual HA in green does just as good as the parental H3 in red at eliciting immune responses. H1 vaccination and mock infecting give you no response. So not only can we pack twice as much hemagglutinin on this virus particle, but these antigens can be seen and efficiently, and immune responses can be efficiently generated against them. Next slide. But in order to determine kind of the functionality of this, we can challenge. And on the left is a PR8 H1N1 challenge. So again, if we vaccinate with PR8 as a control in black, all the animals are protected. Our dual HA virus in green also protects animals from challenge. But if you challenge animals that are similarly vaccinated with an H3N2, the H1N1 no longer protects, but our dual HA virus does. So again, we can make viruses with HAs, they're immunogenic, and the immune responses that they elicit are also protective. Next slide. So I'll wrap up in case there are any questions. We'll leave some time for that. In summary, the idea is influenza virus particles, vaccinating with influenza virus particles do some things well, but that protection is limited and matched to matched or closely related strains, and we need to improve on that. With our kind of subtractive analysis data that I showed you, I think it's clear that the anti-HA immunity is really important for reducing viral titer, but the other viral components together are major contributors to protection from disease. And we've developed and are continuing to develop kind of genetic approaches to change the structural and biophysical characteristics of that virus particle, such that when we vaccinate with them, again, still in the context, of an authentic virus with all the other proteins there, we can change the immune responses, which then have implications for, kind of the degree of the protection that's elicited. And, the final point is that as we continue to make these virus particles with different compositions, we may need to think about influenza vaccines, not only just in terms of being better, but also potentially vaccines for different kind of applications, specialized classes of influenza vaccines. And I will leave it at that. Oh yes, the most important slide. The people who actually did the work. So the work at the beginning was done primarily by a postdoc in the lab, Zhao Chen, and the stuff I showed you at the end with the genetic approaches was done by Al Harding, and of course our funding from these sources. We couldn't have done the work without the money to do it. So thank you. Now I will take questions.
All right. Thanks, Nick. There was one question in the chat. A very interesting approach to new vaccine candidates. Do these novel platforms grow and replicate well in vaccine cell lines or eggs?
Yeah, right. That's a great and important question, right? There's a feasibility of manufacture question. So the answer is it's complicated. When we reduce hemagglutinin, the viral titer, viral yields are reduced about tenfold for that approach. When we increase hemagglutinin, those viruses actually replicate to the same level in both eggs and MDCK cells as the parental kind of wild type strains. And we're also comparing that to this background PR8, which is a background used to generate influenza vaccines. So it's a very robust grower. So that's the long answer to your question. Yeah, they grow to similar levels.
Great. Thanks, Nick. I don't see any additional questions in the chat, and I think in the interest of time, I will turn it back over to Ted so he can introduce the next session chair.